Hi, today I'm going to be going over Module 1. So starting with Chapter 1, what is anatomy and physiology? So anatomy is the study of the body's structures, and there are many branches under that, including gross anatomy, which is studying larger structures, for example, maybe the brain. Microscopic anatomy is the study of structures that can only be seen with a microscope, like cells and tissues. Regional anatomy is studying the interrelationships of all the structures in a specific body region. Systemic anatomy is studying the structures that work together to perform a specific body function. And for physiology, it's the study of the chemistry and physics within the structures of the body and how they work together to support life. And the branches under that is homeostasis, which is when our bodies try to maintain a consistent and comfortable internal state for our well-being. Now, the structural organization of the body includes atoms, organelles, cell, tissue, organ, and organ system. So atoms are the smallest building block of matter, and it's the simplest level, and it includes the chemical level, which makes then molecules. Organelles are flexible membranes that enclose cytoplasm, and they're water-based cellular fluid together with a variety of tiny functioning units. Cells are the smallest working group of a living organism, and tissues are a group of many cells that work together to perform certain functions and jobs in the body. An organ is a distinct structure of the body composed of two or more tissue types, and an organ system is a group of organs that work together to do big functions to meet the physiological needs of the body. The functional characteristics of human life start with metabolism, which is the basic function that us as organisms need to consume energy and molecules from the foods that we eat, which then gets converted into fuel to move, sustain body functions, and build and maintain the body's structures. The body's able to do this through two things, through anabolism and catabolism. So anabolism is when smaller molecules combine into larger complex substances, and by using energy, the body can put together the complex chemicals it needs by combining small molecules from foods that we do eat. And catabolism is when larger substances are broken down to smaller molecules. And this reaction is able to release energy so that the body can use it to make up the structures and substances that we need for life. Now, the next one is adenosine triphosphate, which is ATP, as we usually say. So every cell uses ATP to store and release the energy, and the process uses both anabolism and catabolism. Responsiveness is how an organism can adjust to changes in an external and internal environment. For example, we would move towards food and a water source, and we will not go towards perceived dangers. Movement is the way that the joint and individual organs and cells move. Everything inside the body is constantly moving to maintain what it needs to survive. Development is the changes that the body goes through, and this can include cell differentiation when unspecialized cells become specialized. Growth is the increase in the body's size by increasing the number of existing cells and non-cellular material. And reproduction, which is to make new organisms from parent organisms by the male and female reproductive systems. The 4 requirements for human survival include oxygen, nutrients, narrow range of temperature, and narrow range of atmospheric pressure. So oxygen is essential because the chemical reactions that keep the body alive. Brain cells tend to be the most sensitive to lack of oxygen because they need a lot of constant ATP. Nutrients are found in the food and drinks that we have, and the 3 basic classes are water, the energy-yielding and body-building nutrients, and micronutrients, which is like vitamins and minerals. Narrow range of temp is the need for the body to stay at a temperature that it's comfortable at, which is 37 degrees Celsius or 98.6 degrees Fahrenheit, because the body isn't able to do the necessary chemical reactions if it is too far out of this temperature. And the narrow range of atmospheric pressure is the pressure that is given by the mix of gases, mostly nitrogen and oxygen, in the Earth's atmosphere. It keeps the bodily gases dissolved. Without the gas in our blood would expand, and that could block blood vessels and make the cells break. Homeostasis is very important for human functioning. What is it though? It's when the body tries to maintain and monitor a consistent and comfortable internal state for our well-being, and all physiological conditions have a particular set point, which is like the normal range. And it's important because it brings our body back to normal range when it gets out of it, which keeps us healthy and not sick. So this is the anatomical terminology that is usually shown and known. So the anatomical position means the standard body map that this is. And the body is standing upright, the feet at shoulder width apart, toes forward, and arms at side. So these are the directional terms of that, which is superior inferior, cranioclaudal, posterior or dorsal, and anterior or ventral. And we obviously have our right and left, which we also know that one. And proximal distal, medial lateral, and body planes, which is sagittal, which is cutting it sideways. So each side is cut. And then frontal, which is front to back. And transversal is right through the middle, flat through the middle. And the body cavities include these colors, which is the cranial and vertebral cavity, which both of those together are the dorsal cavity. And then we have our thoracic cavity, which includes superior mediastinum, pleural cavity, pericardial cavity within there, and the diaphragm, and then the abdominal cavity, which is the biggest one, and the pelvic cavity. And then we see the ventral body cavity is all of these together. And then the regions and quadrants of the body of the diaphragm include the right hypochondriac region, the epigastric region, and the left hypochondriac region. And then going down the middle, the right lumbar region, the umbilical region, and the left lumbar region. And then all the way at the bottom is the right iliac region, the hypogastric region, and the left iliac region. And in our quadrants, we have right upper quadrant, right lower quadrant, left upper quadrant, and left lower quadrant. Next, the medical imaging techniques that are used in medicine very commonly include the X-ray, which X-rays use high-energy electromagnetic radiation with short wavelengths capable of penetrating solids, and they ionize gases. They are slightly embedded by soft tissues, which show up as gray on the X-ray plate, which is like the skin and fleshy part. Whereas hard tissues like bone, they block the rays, producing a light-toned shadow. They're commonly used to view damaged bones and teeth, but however, the radiation can potentially damage cells, which can lead to cancer in the future, possibly. Next is computed tomography, which is CT scans, and they use computers to analyze several cross-sectional X-rays in order to reveal minute details about structures in the body. This is able to rotate 360 degrees to take images similar to an X-ray, however, it is non-invasive. And magnetic resonance imaging, which is MRIs, work because matter exposed to magnetic fields and radio waves were found to emit radio signals. These are non-invasive, but however, they unfortunately cost more. So patients can also be uncomfortable if they are claustrophobic, and they have strong electromagnets. Next is the PET scan, which uses radiopharmaceuticals, substances that emit radiation that are short-lived, therefore it's relatively safe to use on the body. The main benefit is that it can illustrate physiologic activity, including nutrient metabolism and blood flow of the organs or organs being targeted, whereas CT and MRI scans can only show static images. And lastly, ultrasonography, which ultrasounds use the transmission of high-frequency sound waves into the body to be able to generate an echo signal that is converted by a computer into real-time images. It's the least invasive of all of these imaging techniques, but the disadvantage is that the image quality depends heavily on the operator, and it's not able to go through bone and gas. So now starting with chapter 2, starts with the fundamental composition of matter, which means matter is anything that takes up space and has mass, and all matter is made up of one or more of the 92 elements. The 4 most abundant elements in the body include carbon, hydrogen, oxygen, and nitrogen, which can be shortened as CHON to remember. And the 3 subatomic particles are proton, neutron, and electron, which atoms are made up of all of these. The relationship between an atom's number of electrons and its relative stability is that all atoms have electron shells, and an electron shell is a layer of electrons that circle around the nucleus at a distinct energy level, and then a valence shell is an atom's outermost electron shell. If the valence shell is full, the atom is stable, which means the electrons are unlikely to be pulled away from the nucleus by the electrical charge of other atoms. If the valence shell is not full, the atom will be reactive. So the types of bonds are ionic bonds, covalent bonds, and hydrogen bonds. So ionic bonds are formed by the attraction of a cation's positive charge and an anion's negative charge. So in this picture, the sodium commonly donates an electron to chlorine, becoming the cation Na+. When chlorine accepts the electron, it becomes the chloride anion Cl-. With their opposing charges, these 2 ions are able to strongly attach to each other. Covalent bonds are when they share electrons in a mutually stabilizing relationship, and covalent bonds are stronger than ionic bonds. Hydrogen bonds are usually formed when a weak positive hydrogen atom is already bonded to one electronegative atom. Hydrogen bonds always include hydrogen that is already part of a polar molecule. So, energy and chemical reactions include kinetic energy, which powers any type of energy in motion. Potential energy is energy matter possesses because of the positioning or structure of what it's made of. Chemical energy is the form of potential energy in which energy is stored in chemical bonds. When those bonds are formed, chemical energy is invested. When they break, chemical energy is released. Mechanical energy is stored in physical systems like machines or the body. It directly moves the movement of matter. It directly powers the movement of matter. And your muscles provide mechanical energy to be able to move objects. Radiant energy is when energy is emitted and transmitted as waves rather than matter. These waves vary in length, and the full spectrum of radiant energy is called the electromagnetic spectrum. The body uses the ultraviolet energy of sunlight to convert a compound in skin cells to vitamin D. And the human eye evolved to see these wavelengths, which is the color of the rainbow. And electrical energy is energy supplied by electrolytes in cells and body fluids, and it contributes to the voltage change that helps transmit impulses in nerve and muscle cells. The importance of organic compounds is that organic compounds are substances that don't contain both carbon and hydrogen. Many inorganic compounds do contain hydrogen atoms like water, H2O, and hydrochloric acid produced by your stomach. Water is a large part of many of the body's lubricating fluids, like synovial fluids in the joints, keeping abdominal organs friction-free, and keep food moving through the digestive. But water also protects cells and organs from physical trauma. And a heat sink is a substance or object that absorbs and dissipates heat, but does not experience a corresponding increase in temperature. In the body, the water absorbs the heat generated by chemical reactions without greatly increasing in temperature. If the body's too cold or too warm, the water helps that. And a mixture is a combination of two or more substances, each of which has its own chemical identity. The air we breathe is a gaseous mixture containing three elements, nitrogen, oxygen, and argon, and one compound, carbon dioxide. There are three types of liquid mixtures, and they all have water as its main component. For cells in the body to survive, they must be kept in a water-based solution. And next, salts dissociate into ions, and these ions are electrolytes which are capable of conducting an electrical current in solution. This property is necessary and critical to the function of ions in transmitting nerve impulses and prompting muscle contraction. Other salts are also important to help break down dietary fats and calcium phosphate salts for teeth and bones. And acids are a substance that releases hydrogen ions in solutions. Strong acids are compounds that release all of their hydrogen in solution, which means they ionize completely. Hydrochloric acid, which is released from the cells in the lining of the stomach, is a strong acid because of its hydrogen in the stomach's watery environment. The strong acid is necessary in helping the digestion and kills the ingested microbes. And bases are a substance that releases hydroxyl ions in solution, or one that accepts hydrogen already present in solution. Strong bases release most of their hydroxyl ions. Weak bases release only some of these. Food mixed with hypochloric acid from the stomach would burn the small intestine if there was not a release of bicarbonate. So, to compare and contrast these four classes, we have first proteins, which they are made out of carbon, hydrogen, oxygen, and nitrogen. They're organic molecules of amino acids linked by peptide bonds, which includes keratin in the epidermis, collagen in the dermis and bones, and the meninges covering the brain and spinal cord. And they play a role in the digestive enzymes, antibodies, and neurotransmitters. Carbohydrates are made out of carbon, hydrogen, and oxygen. They are also known as saccharides, which means sugar, and all body cells need sugar to be broken down into energy. Lipids are made out of carbon, hydrogen, and oxygen, and they are fatty, waxy, or oily compounds that are essential to many human body functions and serve as the building blocks for living cells. Nucleic acids are made of long chain-like molecules called nucleotides, which have three main parts, and they are naturally occurring chemical compounds found in all cells and are a vital part to any living organism. In Chapter 3, we have the cell membrane, which is made mostly of back-to-back phospholipids, various proteins, and cholesterol, which contributes to the fluidity of the membrane. So the phosphate group is negatively charged, making the head polar and hydrophilic, which means attracted to water, and the lipid tails are uncharged, nonpolar, and are hydrophobic, which means repels water. And since some lipid tails consist of saturated fatty acids and some contain unsaturated fatty acids, it makes them empathetic, which a molecule has both a hydrophilic and hydrophobic region. The cell membrane can also regulate the concentration of substances inside the cell. This causes the membrane to be selectively permeable, meaning it only allows substances that meet what it wants to pass through. And they also can move through the membrane. They either do it by passive or active transport. Passive transport is moving of substances across the membrane without the use of cellular energy from ATP. Active transport is the movement of substances across the membrane, then using energy from ATP. So the functions of the cytoplasmic organelles are the endoplasmic reticulum, ER. It provides passages throughout much of the cell that function in transporting, synthesizing, and storing materials. The winding surface of the structure results in large membrane surface to support itself. But there is two types of this. There is rough ER and smooth ER. Rough is covered in ribosomes that work as the site of the protein synthesis, which is all the little dots. And it wraps around the mRNA to start the process of translation, then to become protein synthesis. And smooth synthesizes phospholipids, the main component of biological membranes, as well as steroid hormones. The Golgi apparatus is responsible for sorting and modifying and shipping off the products that come through the rough ER These products are sorted through the apparatus and then are released at the opposite side after becoming new vesicles. Lysosomes are organelles that contain enzymes that break down and digest unneeded cellular components such as damaged organelle, and they're important for breaking down foreign materials. So the nucleus is surrounded by a membrane called the nuclear envelope, which consists of two adjacent lipid bilayers and then a thin fluid space in between. The bilayers are across them are nuclear pores, which are tiny pathways for proteins, RNA, and solutes between the nucleus and cytoplasm. And the nucleolus is a region of the nucleus that is responsible for manufacturing the RNA necessary for making ribosomes. Within the nucleus are threads of chromatin, composed of DNA and associated proteins. Along these threads, the DNA is wrapped around in a set of histone proteins. A nucleosome is a single wrapped DNA histone complex, and the chromosome is composed of DNA and proteins. It is a condensed form of chromatin. So DNA replication has 3 steps, and step 1 being initiation, where the 2 strands are able to unzip themselves and special enzymes like helicase untwist and separate the 2 strands of the DNA. Step 2, elongation, where each strand becomes a template where the new complementary strand is built, and DNA polymerase brings in the correct bases to connect to the template strand, synthesizing a new strand. Step 3, termination, is the 2 original strands are connected to their own and finished strands. DNA replication is now complete, and there are 2 new identical DNA molecules. So to get from DNA to protein, you have to start with transcription, where you still have the same 3 steps of starting with initiation. Transcription is a synthesis of a strand of mRNA that is complementary to the gene of interest. So step 1, initiation, a region at the beginning of the gene, called a promoter, triggers the start of transcription. Elongation is when its transcription starts, when RNA polymerase unwinds the DNA. The coding strand becomes the gene template, and the polymerase then aligns the correct nucleic acid, either A, C, G, or U, with its complementary base on the coding strand. RNA polymerase is an enzyme that adds new nucleotides to a growing strand of RNA, and this process builds a strand of mRNA. Lastly, step 3 of termination, a sequence of nucleotides called the terminator sequence, causes a new RNA to close up on himself. This causes the RNA to separate from the gene and from RNA polymerase, therefore ending transcription. And lastly, for translation, the next big step is the process of synthesizing a chain of amino acids called a polypeptide. It first needs a translator, which is a molecule that will conduct the translation, and second, a substrate on which the mRNA strand is translated into a new protein. Ribosomal RNA, rRNA, is when together with proteins they make a structure of ribosome. When the mRNA molecule is ready to be translated, the 2 subunits are able to come together and attach to the mRNA, and the ribosome provides a substrate for translation, bringing together and aligning the mRNA molecule with the molecular translators that must decipher its code. Transfer RNA, tRNA, is a type of RNA that varies the appropriate corresponding amino acids to the ribosome and can attach a new amino acid to the last, building the polypeptide chain one by one. tRNA transfers a specific amino acid from the cytoplasm to a growing polypeptide, and the tRNA molecules must be able to recognize the codons on RNA and match them with the correct amino acid. The sequence basis on the molecule is called an anicodon. Now with the steps in cell division, they start with prophase, then we have prometaphase, and metaphase, anaphase, telophase, and then the final result is cytokinesis, where we get the 2 cells. And the other type of cell division is meiosis, which it has 1 and 2. So the DNA is replicated this is the way that we create the somatic cells. For a cell to differentiate or become more specialized, it will probably need to do big changes in its shape, size, and metabolic ability. and overall function. All cells have the same complement of DNA, but each cell only reads their own part of DNA for their own use. Technically, this is called the unique genetic expression of each cell, and for each cell to differentiate, it needs to manipulate the genes that will be expressed, and the main way that genes are turned on or off is through transcription. And it is a class of proteins that bind to specific genes of the DNA molecule to promote or inhibit it. And lastly, in Chapter 3 is going over some of the representative cell types, which starts with epithelial, and they protect the body and secrete fluids. They are part of the epidermis. They are found by lining the GI tract organs and hollow organs, and they have no direct blood supply. Now for connective, they support, protect, and connect the other tissues. They have specialized cells and ground substance, along with special fibers like collagen, keratin, and elastic. Muscular, they allow movement for expanding and contracting. They are excitable, so they're able to respond to a stimulus, and some are voluntary and some involuntary. And lastly, nervous, which send and receive electrochemical signals throughout the body, and they're made of two cell groups, neurons and neuroglia. Thank you. That is the last thing we have to talk about. Have a good day.